Vanakkam. Welcome to this course on biomedical instrumentation. In the previous set of videos, we looked at electrode theory in very good detail. From this video onwards, we will move forward to specific methods or specific use of these recording systems. We will start our discussion with bioelectricity. Actually, we did discuss this in previous set of videos briefly. So, we discussed the foundations of these. For example, we have discussed the sodium potassium pump, what causes the steady potential difference across the excitable cell membrane, and we discussed action potential and its propagation. Okay, we discussed action potential, but maybe not its propagation. Okay, that is fine. We discussed action potential and what is it that we are interested in measuring. This. So, in this video, we will discuss electricity and the human body and bioelectric potentials of interest for us. Essentially, the connection between electricity and the human body was first discovered by Luigi Galvani. This picture is given here, this, who showed that muscles and nerves produce electrical charge or electrical change. When you apply electricity, there is a twitching of the muscle that happens. So, while experimenting with frog legs for a long time, this field was called as the frog science. Okay, a lot of these studies on muscles especially involves frog sartorius muscles. Okay, this is called as a science of the frog. Everybody who studies uses frogs as the model system, model animal for their this. So, for a long time this was the frog science. Okay, so while he was experimenting with frogs, he observed that these muscles twitched when a metal probe touched the nerve. This was groundbreaking because until then it was not thought that living tissues could produce electrical signals naturally, because before that it was thought life processes or biological processes were entirely mechanical or entirely biochemical or chemical oriented. This discovery of the twitching of the muscles in frogs essentially became the foundation of what later came to be found as the bioelectricity. It is the field that studies the electrical properties of biological cells and tissues, in particular the excitable cells. Bioelectricity in excitable cells. What are excitable cells? Neurons and muscle cells. By the way, that is not to say that the other cells do not have any form of electricity. They do, but not as much. So, they do not have this steady potential difference across their plasma membrane. So, the body produces natural monitoring signals that we can use for our monitoring of health. These are called as bioelectric potentials. They may be recorded, they may be related to nerve conduction of electricity in neurons, right? Neurons, peripheral nerves for example. They may be related to brain activity that you measure, activity, what is happening in somebody's brain, what am I thinking when I am looking at this camera. Is it possible to decode the thoughts inside a person's head? To some extent, yes. For example, there are also these beautiful approaches whereby through using EEG, it is possible to think about what the person is, what music the person is hearing or what music the person is humming inside his head. People are working with such advanced problems through EEG. This is called as a brain activity, electroencephalography, EEG, or related to heart activity or heartbeat, heart activity also called as electrocardiography, ECG, or we are interested in muscle activity of specific skeletal muscles. Heart is also a muscle, but it is not a skeletal muscle, it is a cardiac muscle. So, that muscle activity we call as electromyography or EMG. These three modalities we will look in great detail in the next set of videos. In nerve conduction, electrical signals move through neurons or nerves, and muscle activity, muscles contract and relax as a result of the electrical activity that happens, there is a mechanical activity that follows in the muscles. And these electrical activity are ionic voltages. Remember this, these are not electronic, they do not travel at the speed of light. Absolutely critical to remember this. These are ionic potentials that emerge, that originate from electrochemical actions of the special excitable cells. Special cells means excitable cells. So, our systems to measure this essentially are transducers. Electrodes are transducers that convert these ionic potentials into electronic potentials. We looked at that in good detail in the previous 10-15 videos, right. So, with this we come to the end of this video. Note that life itself is fundamentally electrical, because the sodium potassium ATPase, whose function I discussed in one of the previous week's videos, right, sodium potassium pump, the amount of ATP that is used by the sodium potassium pump, approximately 25% of the total ATP, anywhere between ranges between 20 to 40% of the total ATP is used for functions such as sodium potassium pump, essentially signifying and signaling, giving an idea of nature's commitment to this electrical process. There must be a strong evolutionary reason why this is important. What are those reasons? This we will see in the future set of videos on ECG, EEG and EMG. The quantum of coverage in this week will be limited to about 2.5 hours, and then we will continue in the next week. With this we come to the end of this video. Thank you very much for your attention.